The history of molecular evolution starts in the early 20th century with comparative biochemistry, but the field of molecular evolution came into its own in the 1960s and 1970s, following the rise of molecular biology. The advent of protein sequencing allowed molecular biologists to create phylogenies based on sequence comparison, and to use the differences between homologous sequences as a molecular clock to estimate the time since the last common ancestor. In the late 1960s, the neutral theory of molecular evolution provided a theoretical basis for the molecular clock, though both the clock and the neutral theory were controversial, since most evolutionary biologists held strongly to panselectionism, with natural selection as the only important cause of evolutionary change. After the 1970s, nucleic acid sequencing allowed molecular evolution to reach beyond proteins to highly conserved ribosomal RNA sequences, the foundation of a reconceptualization of the early history of life. Early history. Before the rise of molecular biology in the 1950s and 1960s, a small number of biologists had explored the possibilities of using biochemical differences between species to study evolution. Alfred Sturtevant predicted the existence of chromosomal inversions in 1921, and with Dobzhansky constructed one of the first molecular phylogenies on 17 Drosophila pseudo-obscura strains from the accumulation of chromosomal inversions observed from the hybridization of poly-10 chromosomes. Ernst Baldwin worked extensively on comparative biochemistry beginning in the 1930s, and Marcel Fleurkin pioneered techniques for constructing phylogenies based on molecular and biochemical characters in the 1940s. However, it was not until the 1950s that biologists developed techniques for producing biochemical data for the quantitative study of molecular evolution. The first molecular systematics research was based on immunological assays and protein fingerprinting methods. Alan Boyden, building on immunological methods of George Nuttall, developed new techniques beginning in 1954, and in the early 1960s Curtis Williams and Morris Goodman used immunological comparisons to study primate phylogeny. Others, such as Linus Pauling and his students, applied newly developed combinations of electrophoresis and paper chromatography to proteins subject to partial digestion by digestive enzymes to create unique two-dimensional patterns, allowing fine-grained comparisons of homologous proteins. Beginning in the 1950s, a few naturalists also experimented with molecular approaches, notably Ernst Meier and Charles Sibley. While Meier quickly soured on paper chromatography, Sibley successfully applied electrophoresis to egg white proteins to sort out problems in bird taxonomy. Soon supplemented that with DNA hybridization techniques, the beginning of a long career built on molecular systematics. While such early biochemical techniques found grudging acceptance in the biology community, for the most part they did not impact the main theoretical problems of evolution and population genetics. This would change as molecular biology shed more light on the physical and chemical nature of genes, genetic load, the classical balance controversy, and the measurement of heterozygosity at the time that molecular biology was coming into its own in the 1950s. There was a long-running debate, the classical balance controversy, over the causes of heterosis, the increase in fitness observed when inbred lines are outcrossed. In 1950, James F. Crow offered two different explanations based on the paradox first articulated by J. B. S. Haldane in 1937. The effect of deleterious mutations on the average fitness of a population depends only on the rate of mutations, because more harmful mutations are eliminated more quickly by natural selection, while less harmful mutations remain in the population longer. H. J. Muller dubbed this genetic load. Muller, motivated by his concern about the effects of radiation on human populations, argued that heterosis is primarily the result of deleterious homozygous recessive alleles. the effects of which are masked when separate lines are crossed. This was the dominance hypothesis, part of what Dobzhansky labeled the classical position. Thus, ionizing radiation and the resulting mutations produce considerable genetic load even if death or disease does not occur in the exposed generation. And in the absence of mutation, natural selection will gradually increase the level of homozygosity. Bruce Wallace, working with J. C. King, used the overdominance hypothesis to develop the balance position, which left a larger place for overdominance. In that case, heterosis is simply the result of the increased expression of heterozygote advantage. If overdominant loci are common, then a high level of heterozygosity would result from natural selection, and mutation-induced radiation may in fact facilitate an increase in fitness due to overdominance. Debate continued through 1950s, gradually becoming a central focus of population genetics. A 1958 study of Drosophila by Wallace suggested that radiation-induced mutations increased the viability of previously homozygous flies, providing evidence for heterozygote advantage in the balance position. Wallace estimated that 50% of loci in natural Drosophila populations were heterozygous. Moti Kimura's subsequent mathematical analyses reinforced what Crow had suggested in 1950, that even if overdominant loci are rare, they could be responsible for a disproportionate amount of genetic variability. Accordingly, Kimura and his mentor Crow came down on the side of the classical position. Further collaboration between Crow and Kimura led to the infinite alleles model, which could be used to calculate the number of different alleles expected in a population, based on population size, mutation rate, and whether the mutant alleles were neutral, overdominant, or deleterious. Thus, the infinite alleles model offered a potential way to decide between the classical and balanced positions, if accurate values for the level of heterozygosity could be found. By the mid-1960s, the techniques of biochemistry and molecular biology, in particular protein electrophoresis, provided a way to measure the level of heterozygosity in natural populations, a possible means to resolve the classical balance controversy. In 1963, Jack L. Hubby published an electrophoresis study of protein variation in Drosophila. Soon after, Hubby began collaborating with Richard Lewontin to apply Hubby's method to the classical balance controversy by measuring the proportion of heterozygous loci in natural populations. Their two landmark papers, published in 1966, established a significant level of heterozygosity for Drosophila. However, these findings proved difficult to interpret. Most population geneticists rejected the possibility of widespread neutral mutations. Explanations that did not involve selection were anathema to mainstream evolutionary biology. Hubby and Lewontin also ruled out heterozygote advantage as the main cause because of the segregation load it would entail, though critics argued that the findings actually fit well with overdominance hypothesis. Protein sequences and the molecular clock. While evolutionary biologists were tentatively branching out into molecular biology, molecular biologists were rapidly turning their attention toward evolution. After developing the fundamentals of protein sequencing with insulin between 1951 and 1955, Frederick Sanger and his colleagues had published a limited interspecies comparison of the insulin sequence in 1956. Francis Crick, Charles Sibley and others recognized the potential for using biological sequences to construct phylogenies, though few such sequences were yet available. By the early 1960s, techniques for protein sequencing had advanced to the point that direct comparison of homologous amino acid sequences was feasible. In 1961, Emmanuel Margoliouth and his collaborators completed the sequence for horse cytochrome C, followed in short order by a number of other species. In 1962, Linus Pauling and Emil Zucker-Kandel proposed using the number of differences between homologous protein sequences to estimate the time since divergence. an idea Zucker-Kandel had conceived around 1960 or 1961. This began with Pauling's long-time research focus, hemoglobin, which was being sequenced by Walter Schroeder. The sequences not only supported the accepted vertebrate phylogeny, but also the hypothesis that the different globin chains within a single organism could also be traced to a common ancestral protein. Between 1962 and 1965, Pauling and Zucker-Kandel refined and elaborated this idea, which they dubbed the molecular clock, and Emil L. Smith and Emmanuel Margoliouth expanded the analysis to cytochrome C. Early molecular clock calculations agreed fairly well with established divergence times based on paleontological evidence. However, the essential idea of the molecular clock, that individual proteins evolve at a regular rate independent of a species' morphological evolution, was extremely provocative. The molecular wars. From the early 1960s, molecular biology was increasingly seen as a threat to the traditional core of evolutionary biology. Established evolutionary biologists, particularly Ernst, Meier, Theodosius Dobzhansky and G. Simpson, three of the founders of the modern evolutionary synthesis of the 1930s and 1940s, were extremely skeptical of molecular approaches, especially when it came to the connection to natural selection. Molecular evolution in general, and the molecular clock in particular, offered little basis for exploring evolutionary causation. According to the molecular clock hypothesis, proteins evolved essentially independently of the environmentally determined forces of selection. This was sharply at odds with the pan-selectionism prevalent at the time. Moreover, Pauling, Zucker-Kandel, and other molecular biologists were increasingly bold in asserting the significance of informational macromolecules for all biological processes, including evolution. The struggle between evolutionary biologists and molecular biologists, with each group holding up their discipline as the center of biology as a whole, was later dubbed the Molecular Wars by Edward O. Wilson, who experienced firsthand the domination of his biology department by young molecular biologists in the late 1950s and the 1960s. In 1961, Meier began arguing for a clear distinction between functional biology and evolutionary biology. He argued that both disciplines and individual scientists could be classified on either the functional or evolutionary side, and that the two approaches to biology were complementary. Meier, Dobzhansky, Simpson and others used this distinction to argue for the continued relevance of organismal biology, which was rapidly losing ground to molecular biology and related disciplines in the competition for funding and university support. It was in that context that Dobzhansky first published his famous statement, "Nothing in biology makes sense except in the light of evolution," in a 1964 paper affirming the importance of organismal biology in the face of the molecular threat. Dobzhansky characterized the molecular disciplines as Cartesian, and organismal disciplines as Darwinian. Meier and Simpson attended many of the early conferences where molecular evolution was discussed, critiquing what they saw as the overly simplistic approaches of the molecular clock. The molecular clock, based on uniform rates of genetic change driven by random mutations and drift, seemed incompatible with the varying rates of evolution and environmentally driven adaptive processes that were among the key developments of the evolutionary synthesis. At the 1962 Wenner-Gren Conference, the 1964 Colloquium on the Evolution of Blood Proteins in Bruges, Belgium, and the 1964 Conference on Evolving Genes and Proteins at Rutgers University, they engaged directly with the molecular biologists and biochemists, hoping to maintain the central place of Darwinian explanations in evolution as its study spread to new fields. Gene-centered view of evolution, though not directly related to molecular evolution, The mid-1960s also saw the rise of the gene-centered view of evolution, spurred by George C. Williams's adaptation and natural selection. Debate over units of selection, particularly the controversy over group selection, led to increased focus on individual genes as the theoretical basis for evolution. However, the increased focus on genes did not mean a focus on molecular evolution. In fact, the adaptationism promoted by Williams and other evolutionary theories further marginalized the apparently non-adaptive changes studied by molecular evolutionists.